Hello and welcome to the second video of the cell biology series. If you haven't watched the first one on the introduction to cells, I'd recommend watching that one first. But without further ado, we get to the second topic, the ultrastructure of cells. And for that, I'll firstly talk about cell organelles in general, and then the cell organelles in eukaryotic cells and prokaryotic cells. And at the end of that topic, we also have two tables, one comparing eukaryotic and prokaryotic cells, and one comparing to eukaryotic cells, so plant cells and animal cells. So you have a summary to that. And then lastly, we have a bit of a deviation. I'll be talking about a short excerpt of the history of microscopy. So if that interests you, enjoy. And we'll be starting with the nucleus, which is the biggest part of the cell, or the biggest organelle within the cell, and it contains the chromosomes. The chromosomes are composed of DNA and histone protein, forming chromatin. And the nucleus is also surrounded by a double-layered membrane that's called the nuclear envelope that has pores for material channels, where material can go in and out of the nucleus, so either from the cell plasma into the nucleus or from the nucleus out into the cell. And there's also the dark nucleolus, which is the site for the ribosome production. Moving on, here we have the endoplasmic reticulum that is associated with the nuclear envelope. It's basically a structure of folded membranes that, in case ribosomes attach to the endoplasmic reticulum, it forms the rough ER, the rough endoplasmic reticulum, which is a site for protein synthesis. And the proteins are also collected in so-called cisternae, that are places between the membranes from which they are transported in vesicles to other cell compartments. And there's also the smooth ER, that is the ER without ribosomes attached, and they have many enzymes on the membranes of the smooth ER, and it has different functions depending on the location. For example, one function could be toxin breakdown in the liver, but also it produces phospholipids for construction of membranes and lipids in the cell. And here we have four organelles in one side. First, we have the Golgi apparatus that is similar in appearance to the smooth ER, and it processes proteins that comes from the rough ER, collects them, modifies them, and packages them, and releases it in vesicles to other organelles or secretes it out of the cell, which is mostly something that happens in the pancreas, where a lot of happens mostly in the pancreas. So we also have the mitochondria that is surrounded by a double membrane, and if you don't know, is the powerhouse of the cell. It's the site of aerobic respiration, and the inner membrane fold forms so-called cristae to increase the surface for production of ATP. And I just noticed that I forgot to talk about what ribosomes are doing, so I'll just say it now. Ribosomes are the site of protein synthesis, either when they're free within the cell plasma or when they're attached to the rough endoplasmic reticulum. And they're made up of RNA, protein, and are without a membrane. Eukaryotic cells have 80S ribosomes and prokaryotic cells have 70S ribosomes, but I'll talk about what that exactly means later on. So lysosome is something like the waste organelle of the cell. So they have an internal structure that has been made by the Golgi apparatus, and it contains hydrolytic enzymes for components of the cell, and it contributes to cell death and breaking down of old organelles. So it basically clears out all the unnecessary and all the old materials within the cell. And we also have the plasma membrane that controls movements in and out of the cell and provides a medium for metabolic reactions. And on the next slide, I have 3 organelles that are exclusive to plants, although there are also other differences. But first of all, these organelles, as I already said, are specific to plants, and the first of which is chloroplasts. And chloroplasts are not even necessarily found in all of the chloroplasts, only in those that are exposed to light and produce chlorophyll, the green pigment. Because of that, it's the site of photosynthesis and has a double membrane and own DNA as well. I believe the mitochondria also has an own DNA. It has own ribosomes and it can reproduce independently. Then we also have the vacuole, and there are some eukaryotic cells where, all of them are eukaryotic cells. What am I talking about? I believe there are some animal cells that also have small vacuoles, but the large ones are specifically in plants. So vacuoles contain water, salt, and add stability because of the pressure on the membrane. Because it is so big, it adds pressure on the cell plasma outside, and that in turn creates pressure on the membrane and cell walls. So it adds to the stability and the structure of the plant cell in and of itself. And it also contributes to the water balance within the cell. And last we have the cell wall. That's composed of cellulose and carbohydrates and supports and structures so that the plant holds an optimum position to catch light for photosynthesis. Moving on the right hand side you can see a prokaryotic cell with things like the plasmid that you can see inside down here. And the plasmid can contain additional small circles of DNA. It replicates independently and it may pass from one cell to the other. And also things like pilus or pili are in some prokaryotes and can connect to other bacteria for exchange of genetic material. And the same goes for flagellum or flagella. That is not in all prokaryotic cells, but in some. It extends out of the cell wall, as you can see here, and it's there for movement. The nucleoid DNA is naked, as you can see here, so there's no nuclear envelope, as opposed to eukaryotic cells that have, and it contains the chromosomes, and it also lacks proteins. And yes, in this structure you can see some of the cell organelles we already talked about. And yes, specific to prokaryotic cells is that it has no real nucleus, only a nucleoid. That is also why it is called a prokaryotic cell, because in Greek it means before the nucleus. It has mostly no organelles and no internal structure. That says it is not membrane bound. And prokaryotic cells are relatively small, so from 0.5 to 10 micrometers for single cells for most prokaryotes. And the cell wall, although they have a cell wall, it's different to that of most eukaryotes. As you can see here, for example, it has a capsule. I already talked about the singular circular chromosome of the naked DNA, and as you can see, it has a simpler cell structure without compartmentalization, and it's also, the prokaryotic cells are bacterial. And here is what I can say to the ribosome sizes that I mentioned earlier. The unit S is used to measure the size of ribosomes. That's called the Svedberg unit. and it measures the behavior of particles during sedimentation, so the different times to sediment when they're being centrifuged, also called the sedimentation coefficient. And that is not of that much importance. You just need to know that when there's, for example, in prokaryotes, I believe it's 70S, that's a unit for, well, to some extent, the size of ribosomes, and they're smaller than in eukaryotes. And additionally, prokaryotes are mostly of the so-called Monera Kingdom, and there are two parts to the Monera Kingdom, the Archaeobacteria and the Eubacteria. The Archaeobacteria are mostly found in extreme environments, for example, volcanoes, I believe, or the deep sea. And Eubacteria are traditional bacteria that include most pathogenic forms, as we know, for example, E. coli, perhaps. Additionally, something that is special to prokaryotic cells is the binary fission in prokaryotes, and that's basically the division of the parent body into two fairly equal parts to produce two identical cells. And this is a form of asexual reproduction that is made by most prokaryotes, although also some eukaryotic organelles. So not only prokaryotes do this, but perhaps chloroplasts also do binary fission, as they can reproduce independently from the cell. If you can remember, I said that earlier. And there's something like a generation time that's referred to, and this means the time required for bacterial cells to double. And the process includes the fact that this DNA is replicated and copies attach to different part of the plasma membrane. The cell pulls apart, and the replicated and the original chromosomes are separated. So this reproduction process goes on without mitosis and without cytokinesis. And here you can also see the process in a graphical way. So you see the replication of the genetic material, and there's a cross-wall forming that then divides the two cells. So we have the original and the reproduced. And that is the process of binary fission, as here ingrowing cell walls meet and two identical cells are formed. Moving on to animal cells. They are more complex than, all eukaryotic cells are more complex than plant cells, all eukaryotic cells are more complex than prokaryotic cells, and animal cells, for example, have no cell wall, have no vacuole, have no chloroplast. Those are specific to plant cells, and as you can see, they include all of the cell organelles I already talked about, arranged in a graph. Some additional ones that are specific that I have not mentioned, for example, the secretory vesicles and the peroxisome. But in general, you can find in different graphs different cell organelles, but the main ones, such as the mitochondria, the nucleus, and the Golgi apparatus, and so on, are always found there. And here we also have a picture of the plant cell, and here are the aforementioned specific vacuole, chloroplast, and cell wall. And there's also somewhere a tight tight junction was in animal cells. I'm sorry. Tight junctions are also, for example, found in this here animal cell that are impermeable to, that are a form of impermeable junction and that bind cells together. And something I haven't mentioned is that, for example, the centriole here is a microtubular and is therefore nuclear division. But other than that, I've already talked about the specific cell organelles in the beginning. And here you can see the differences in a tabular form between eukaryotic and prokaryotic things. So on the left-hand side, you can see the structure and the occurrence in the two roots, and the nucleus is usually present in eukaryotic cells. They also contain a nuclear envelope, contain chromosomes and chromatin, whereas in prokaryotic cells, as you saw before, they have no nucleus, and therefore there is no nuclear envelope and no nucleolus. Then we have mitochondria that's usually present in eukaryotic cells and never present in prokaryotic cells. We have chloroplasts that are present in some plant cells but never in prokaryotic cells. We have the ER that is usually present in eukaryotic cells and never present in prokaryotic. We have ribosomes that are relatively large with a 30 nanometer diameter, so about 80 S, the Svedberg unit. And in prokaryotic cells, they're in comparison to that relatively small, so they're only 20 nanometers and 70 S. Now, chromosomes in the eukaryotic cell are long strands with histone proteins, and in the prokaryotic cells, the DNA is present but not associated, but a circular may be present. Right. We also have a cell wall that's always present in plant cells for the eukaryotes, made up of cellulose, but it's never in animal cells. In prokaryotes, it's always present, and it's made of peptidoglycan. Peptidoglycan. Sorry. And lastly, something I haven't written down is flagella that you saw in the picture of the prokaryote is sometimes present for movement's sake in eukaryotes, and also some flagella is present in prokaryotes for different structures, but they have different structures than the eukaryotic ones. And now in the last comparison we have the animal cell and the plant cell. And you mostly know this, but this is just to summarize what I've already said. There's no cell wall in animals, there is a cell wall in plants. There are small vacuoles in animal cells and large vacuoles in plant cells. There are no chloroplasts in animals and chloroplasts in plants. There is cholesterol in the plasma membrane in animal cells, and there's no cholesterol in the plasma membrane in plant cells. There are centrioles in the animal cell, there are no centrioles in the plant cell. And then we also have the fact that animal cells store glycogen, whereas plant cells store starch. And you also sometimes see in pictures of plant cells that you have starch molecules or granules within the cell itself. And now we come to a bit of a deviation from what I've already talked about, but in the first video I mentioned the importance of microscopy for the discovery of cells and their functions and structure, and because of that it's perhaps important to look at the history of microscopy in general. So in 1500 there was some things called convex lenses that allowed for the first time a greater magnification than a 5 times magnification. We also have in 1595 a scientist named Zacharias Janssen who made the first compound microscope that looks very primitive to what we have today. It's basically just a tube with some glass in it that magnifies images. In the 1600s we have more progress, so we have in 1662 Robert Hooke, who coined the first germ cell, what I already mentioned in my first video. In 1675 we have Antony van Leeuwenhoek, that made something like a glorified magnifying glass, so it was a single lens microscope. And then in the 1800s it becomes interesting because we have this thing called chromatic aberration, the discovery and the use of the fact that blue light refracts more than red light, and that creates a focal point when using glass. And the same thing was then further developed by Joseph Jackson Lister in 1830. When he used spherical aberration through focusing through a curved lens, where the more light or the more pictures there were on the edge of the lens, it focused clearer. In 1878, we have Ernst Abbe that made the formula for correlating resolution to wavelength to light, so that they could calculate the maximum resolution of the light microscope. And in 1903, we have Richard Zimondi that invented the ultra microscope that could actually see things that are smaller than the wavelength of light. In 1932, we have Fritz Zernike that made a phase contrast microscope for transparent or colorless objects. And in 1938, Ernst Rustiker, who made the aforementioned, well, in the last video I mentioned it, the TEM, the transmission electron microscope, that could see through electrons really small images, or magnify really small images because of the shorter wavelength of electrons as opposed to a light microscope. And in that same year, I believe, there was Manfred von Ardenne who made the SEM, the scanning electron microscope, who enabled those small electron magnifications to be imaged, so that an image could be restored from what has been seen by the TEM. And lastly, what I wanted to talk about are in 1981, Gerd Binning and Heinrich Rohrer made the so-called STM, the scanning tunneling microscope, that made it possible for 3D images at an atomic level to be magnified. And you can see in a relatively short number of years after the single lens microscope how much development and improvement there has been, that we can now project 3D pictures of things on an atomic level. And yes, that also greatly helped with the further discoveries and development of cell biology. And with that, these are my references. I'm ending the second topic of cell biology. As you can see here, my sources. And thank you for watching. I hope you have a nice day.